My name is Komatsu, and I'm from the Smithsonian Environmental Research Center. And I'm working in this project to look at the role of soil microbial communities in driving soil health and potential consequences for above-ground plant material. So we are looking at extracellular enzyme activity, so what these microbes are putting out into the soil and how that functions to decompose plant litter and cycle nutrients through the system, as well as looking at the composition, so the identities of the microbes that are in these soil communities and how they might be functioning in this system. So we're doing that across our different grazing treatments as well as our different rainfall manipulation treatments to see, does drought affect these microbial communities and how they're functioning, as well as does the different grazing pressures affect microbial composition. So we're looking at 3 different grazing treatments to think about different options that ranchers in this region could utilize under conditions of drought. So we're trying to see what the better options could be of these 3 realistic scenarios that we're looking at. One that mimics destocking during a drought condition, one that mimics continuing heavy grazing, and then one that mimics rest during recovery. So trying to determine what of these options are the best for this system. And then we can work with these ranchers again who have given us these suggestions for our treatments to try and help them figure out what they can do in their land under these multi-year drought scenarios. The way that we're doing these grazing treatments is by bringing cattle into these large blocks that we've established. So the way that we've established these grazing treatments is to split our large blocks up into 3 different paddocks where we're going to introduce cattle and allow them to graze until they've grazed the area down to either a heavy usage, a moderate usage, or a light usage. And we could split up our blocks using cattle panels to separate out the cattle from these different areas to get the grazing intensity that we're interested in. And then after they graze, we remove the cattle and we can come back through and measure our plant community composition, our soil microbes, as well as below ground root production.